right. Hi, everybody. I hope that you are all doing okay and having a great week. Hopefully the projector will actually load in a second. Remember that we have the PRR assignment due on Friday. Today we are going to talk about T-cell activation. And really we're going to be thinking about a lot of signaling events. So again, just to kind of orient you, really our goal here is thinking about what's happening early after encounter with antigen or early after infection, in order to start our adaptive immune response. And we're thinking about kind of some of these early events that are happening to allow us to have a large antibody or T-cell response. And so we're now kind of in this idea of where we're kind of having these cells start, particularly T-cells, actually recognize that antigen and become activated. I've introduced you to this view of thinking about T cells in the periphery, and we'll be seeing this view a few times. So right now, today, we're largely going to be focused on the T cell making its initial contact with the antigen presenting cell. So the T cell at the beginning of this is a naive CD4 or CD8 cell. It has not yet been activated. And through the processes that we're going to see, that cell is going to become an activated cell. It's going to undergo some rounds of proliferation, and it's also going to differentiate to gain the functions that we see here at the end. And pretty much our whole time today, and say half of our next class period, we'll be thinking about kind of the cell getting activated and going from being a naive cell to a cell that now has gained its effector function and now can do something about the microbes. And then the second half of our next class and the class after that, we're going to talk about the effector functions. So note that we're sort of in this left-hand part of this slide for right now. And this sort of expands some early events into a few different steps. So note that we're kind of going to be a little across here in some certain places. Really what we're going to be thinking about here is what happens when the MHC plus peptide interacts with the T cell receptor. Remember, the T cell receptor includes both the T cell receptor sort of main chains, particularly the alpha and beta chains that are made during VDJ recombination, as well as the co-receptors, CD3. And we talked about the importance of the ITAM in the cytoplasmic domain of CD3. And the co-receptor, either CD4 or CD8, that will be binding to the MHC. So a big part of what we're going to talk about today is what happens in the T cell as a result of this interaction in order to help the T cell get activated. And so in that sense, we're really kind of zoomed in on this interaction that is being shown right here. Just as a reminder, and this will come up much more a little bit later today and much more on Friday. In reality, in order to have a naive T cell, so a T cell that has never previously been activated, in order to get that cell activated the first time, we need a few different signals coming together in that cell. Some immunologists will call them signal 1 and signal 2. Some people even call the other one signal 3. That one's like signal 1 and signal 2 is very standard immunology terms, whether you call this one signal 3 or not. Different people do different things. And so we're going to have this signal from the T cell receptor known as signal 1. That's important. But there's also this second signal, this co-stimulatory interaction that I will mention a little bit later on today, and I'll talk about much more on Friday, that is really critical for making this actually work. And so know that today, most of our focus is going to be on signal 1. Signal 2 is also very important. I mentioned that sometimes people also talk about signal 3 in turning on the T cell. The thing that we mean when we talk about signal 3 is really that cytokines are also important. And exactly which cytokine we're talking about can vary in a few different locations. So right now I'm just going to say cytokines. There are a couple different cytokines that are important in a couple different places. And so note that some cytokines are key here. If you go back to this slide, you can again see cytokines being really critical here. We are going to talk a little bit about one of those cytokines today called IL-2, but it is not the only cytokine. So when we talk about IL-2 later today, that will kind of be a part of signal 3, but it's not the only part, and we'll see some other parts of that later on. So like I said, today is really about signal 1, T cell receptor signaling. On the next 2 slides, I have some information from some somewhat recent papers about some of the details of T cell receptor signaling that we're going to be thinking about. So right now, this is 2019, so this is old-fashioned. This is what happens in about the first 10 minutes. What you notice, some of your eyes popped, is that this is pretty complicated. There's a lot going on here. And this is actually a further downstream part of this. What you should notice here is that there's a lot going on. I am not going to tell you every single thing on these diagrams. I'm going to hit some highlights and hit some pretty important themes. And so if you are looking at your textbook and you're like, oh my God, but this protein, she didn't say it. What am I going to do? It's okay. I'm not telling you every single piece here. I'm going over some kind of big picture themes. And one thing that I really like to show you here is that in general, whether we're talking about the T cell receptor signaling or if we were talking about the B cell receptor signaling, there's sort of this set of common themes that happen. And so we're going to be seeing this kind of idea of common themes. And we are largely going to be talking about the signaling events that your textbook talks about in figure 8.11 here. So we could think about all sorts of stuff going on with signal transduction. But one of the things that I think is really important when we think about all of the things that are going to happen with T cell receptor signaling is to think a little bit about the goal. What are we trying to make this T cell do? What is the end goal of the signaling that we're going to talk about? And again, I could go in 1,000,001 directions, but we're going to have kind of a focus goal. So know that this goal I tell you is not the only goal, but it's the only goal we're going to care about. And that goal is that we want the cell, this T cell, to learn how to make the cytokine IL-2. So for a cell that was naive to become activated, that cell needs to make IL-2. So we need to get the right signals to happen in this cell so that it can make IL-2. And so here we can see the IL-2 gene. And remember, our goal is that we can transcribe this gene and then translate the protein and make this cytokine IL-2. And if we look at the promoter of the IL-2 gene, we find that there are binding sites for 3 transcription factors. So in order for the cell to make IL-2, the goal is 3 transcription factors we got to get. One of them is called NFAT, one of them is called AP1, and one of them is called NF-kappa B. So as we look at signaling today, one thing you might think about is this event that we're talking about giving us NF-kappa B? Is it giving us NFAT? Is it giving us AP1? Is it giving us all of those things? So know that everything we're going to be talking about today is going towards the goal of making IL-2. And if your goal is making IL-2, you have to get all three transcription factors. You have to have NF-kappa B, NFAT, and AP1. And so everything we're doing is to get us to that goal. The first step of T-cell receptor signaling is shown here. I've actually already told you a bit about this when we talked about the T-cell receptor, but I'll tell you about it again here. You can also see from the title that this is listed as T-cell receptor proximal signaling. That really means that it's the beginning. It's the things that are happening right close to when the receptor gets a signal. Some other things are going to happen later, but the receptor proximal events are like the ones that happen right at the beginning. One other thing that's really important when I think about receptor proximal signaling is that eventually we're going to get to a point where we're going to have branches in the signaling. And spoiler alert, those branches, one of the branches is going to give us NF-kappa B. One of the branches is going to give us NFAT. One of the branches is going to give us AP1. So we have those three transcription factors we're trying to get to. Well, eventually we're going to have three branches going on. With receptor proximal signaling, we haven't gotten to branches yet. It's all the same and then it's going to branch later. So we're kind of also sort of before the branch point instead of after. So note that we have our T cell with TCR and CD3 on its surface. And we also have our CD4 in this case, or CD8 on the surface of the cell. And these things are diffusing around and are not next to each other. Remember that CD3 has these ITAM motifs in its cytoplasmic domain that contain tyrosines that could get phosphorylated. But we need a kinase to do the phosphorylation. The kinase, and this shows LCK as the kinase. It's often referred to as a member of the SRC family, SRC. But if you told me it was LCK, that's fine. Technically for CD8, it's not LCK, but that's okay. It's LCK's brother. That kinase actually travels with and is sort of interacting very closely with CD4 or CD8. And so normally these things are diffusing separately around the membrane. But if there is an MHC molecule with peptide that comes around, that will bind to both the T cell receptor and CD4 or CD8 and bring those two things that were previously far apart. This thing here on the left is supposed to show them far apart. Kind of hard to draw, to make them fit in the figure but be far apart, so it's not that far. But they're far apart there. They get brought close together because they're both binding to the same thing. And when you bring the extracellular domains close together, that forces the intracellular domains to be close together. And now this kinase, LCK, can add phosphates to our ITAMs. So LCK kinase can phosphorylate the ITAMs, which you can see here as being these new pink dots on the ITAMs. And so this is the first event that happens in T-cell receptor signaling. We're going to see a bunch of different phosphorylation events in different parts of this. And as far as the cell is concerned, the key thing that you might want to think about with phosphorylation events, like phosphorylation of these ITAMs, is that this kind of changes the chemical shape, this changes the charge, this changes some little bits of the chemical structure. And you can imagine that you have now made a new binding site that wasn't there before. So phosphorylated ITAM is like a new binding site. Something new can bind here that could not bind when the ITAM was not phosphorylated. So you suddenly made new binding sites that did not exist previously. If you have new binding sites that did not exist previously, what might you expect would happen? Don't overthink it. Yeah, Josh. So now new partners can bind. You can get some new proteins to come and start acting in this area that weren't able to do so before. And that's exactly what happens. So our new binding sites that were created because of those phosphorylated ITAMs allow a new partner to come in to this process. That new partner is called ZAP-70. And so you can see ZAP-70 here. You can also see ZAP-70 here. And what you can notice about ZAP-70 is that it has these sort of special little binding sites right here that fit onto a phosphorylated ITAM real well. So you can see ZAP-70 didn't have anything to bind to before, but now ZAP-70 has something to bind to because we phosphorylated the ITAMs, so ZAP-70 can come into this location. ZAP-70 is a protein, and it is a protein with a particular function. ZAP-70 is a protein known as a kinase. So if something is a kinase, what does a kinase do? Yep. So a kinase adds phosphates to things. It phosphorylates things. So now we've brought in a new kinase, ZAP-70. And basically what I'm going to tell you is that ZAP-70 phosphorylates stuff. The real answer is that LCK didn't usually phosphorylate all of these ITAMs, and ZAP-70 kind of hits the rest of them. We're not going to have to worry about which one is which. Just ZAP-70 comes in and phosphorylates more partners. Okay? What happens when we phosphorylate these partners, like if they had ITAMs or something? What is this now going to allow? Yep. New binding sites for new partners to come. So what do you think we're going to have happen on the next slide? The next player comes in at this point. At this point, really there are two players that we tend to talk about. I just kind of put them both to say they're both happening here. Exactly what's different between them, don't worry about. You can kind of think of them as relatively interchangeable. One of them is called LAT. The other is called SLP76. And so you can see LAT and SLP76 that are coming in as a result of ZAP-70. And not only that, you can see ZAP-70 kind of pointing to them in an arrow. ZAP-70 can put some phosphates on them. So they're going to get some phosphates now that they came to this area with all these kinases. LAT and SLP76 are two proteins of the same kind of type. They both play the same role in this process. And the role that both of these proteins play is that they are something called an adapter protein. And so what we've done at this point is we have recruited our adapter proteins, and we've put some phosphates on our adapter proteins. And so here you can see SLP76 and it gets some phosphates. Here you can see LAT, gets some phosphates. And the adapter protein step is, in some ways, people find it to be like, blah, lame. And in other ways, it's like crazy, super important. So the first thing I want to tell you about is what the function of an adapter protein is. So remember we just said a few minutes ago that if I call a protein a kinase, I know that its function is to phosphorylate things. Similarly, if I call a protein an adapter protein, there's a function that I'm assuming that it has. And in order to think about that, I want to say something about my South Africa short trek. Or if some of you guys think about international traveling, it would be helpful here. So when I go to South Africa, I have to bring something with me in my suitcase to plug into the wall. What do I have to bring with me to plug into the wall, and why do I bring it? Yeah. Mm-hmm. So I have to bring an outlet adapter, right? And why do I bring that outlet adapter? Yeah. So I want to plug in my devices. Why do I need an outlet adapter to plug in my devices? Yeah. Yeah, because our plugs don't fit. They're like the wrong shape. So like the shape of the outlet there and the shape of the outlet here are different. So if I try to plug in my phone or plug in my laptop, it doesn't fit into their outlet. So I get a little adapter. It plugs into has one side that plugs into their plug, and I have another side that plugs into my laptop, right? And that's really important when I'm traveling internationally. Does that adapter actually do anything functionally? Like, does it mess with voltage or mess with charge or anything like that? No. There are some really special ones that do, but most of them don't. Most of them are just changing the shape from one shape to another, right? And have actually no function themselves. Adapter proteins are just like those plug adapters. They have no function. They aren't an enzyme. They don't actually do anything. All they do is help change the signal from one branch to another. And so what I usually think about in terms of my adapter protein is here is maybe a picture of an adapter protein, and it can get phosphorylated. Here's my adapter protein that's been phosphorylated a bunch of times with the little pink dots at the bottom. And that allows a bunch of other stuff to combine together and start kind of letting the signal go in different directions that we couldn't have done before. But this adapter protein has no enzymatic activity itself. It's just kind of like a site where other things can come and interact with each other to start out changing the types of signals we get. So that's all that the adapter is doing. LAT stands for linker in activated T cells. Linker is basically another word for adapter. I don't remember what ZAP-76 stands for. It doesn't matter. So both of these proteins are adapters. They have no function of their own, but they get phosphorylated, and they just provide so many binding sites for other things to bind so that we can get different branches of our signal to go forward. And so if you remember, I told you that we get to this point where we start to see branching of this pathway in order to get us to our different transcription factors. And so after the adapter is where we start to see the branching. So like everything is the same above the adapter, and then after the adapter, we start to see all of the different branches. So what we're going to be doing in the next few slides is we're going to kind of go from the adapter down a branch until we get to a transcription factor. And then we'll go back up to the adapter and go down another branch. And then we'll go back up to the adapter and go down another branch, okay? So note that we're basically going down the branches here. And I have my branches labeled as I look at them. Basically labeled 1, 2A, and 2B. So you can notice that. So we could call them 2 or 3 branches, however you wanted to say the 1, 2A, and 2B. You'll see as we go why I think about them that way. And the first branch that we're going to follow, which is happening downstream of our adapters, is a branch known as the RAS-MAP kinase branch. This slide talks about the specific details of the RAS-MAP kinase branch. When I learned about RAS-MAP kinase, in an undergraduate class. I learned about it in a slightly different way than it's presented here, and that's still stuck in my mind, so I'm going to explain it that way, and then we'll come back to thinking about this, okay? So if you remember, with our T cell, we're going to have a lot of things happening when that T cell gets activated. And one of the things you saw on that earlier figure was you saw that the cell will do a bunch of cell division, right? You saw a bunch of divisions of the cell. So what would you call a bunch of divisions of the cell? What is the name of that process? Yeah. Proliferation, okay. What if we were in more like a intro class? What would you call that? Probably. When you think about cell division, what word do you often associate with it? Yeah. Mitosis, right? And a thing that makes a cell do mitosis is sometimes called a mitogen. I guess I'll put the mitogen up here. Mitogen. A mitogen leads to mitosis. Okay? So if you put some signal on a cell, like, I don't know, the signal through a T cell receptor, you could call that signal a mitogen. So let's imagine you discovered a protein that gets turned on in a cell when you give that cell a mitogen. So you have a mitogen, you put it on the cell, and the cell makes a new protein. You just discovered this. Excellent job, you. What would you call that protein that is activated by a mitogen? Yeah. You might call it a mitogen-activated protein. Right? Seems like a logical name. And you could even say that's called MAP. And the people who actually discovered this, this is kind of where they got some of these terms from. The thing that we now know is that MAP is a transcription factor. There are many specific examples of a MAP. There's no protein that's actually called MAP. I'm just kind of describing like a class of proteins, and everything we now know in that class are transcription factors. So I could give you names of specific proteins that fall into this class of proteins that are MAPs. I will tell you about them a little bit later. But now just imagine MAP as like a general kind of protein. Okay? So, when the MAPs were described, one of the things that the scientists learned about those MAPs is that those MAPs need to get phosphorylated to become active. So they're off normally, and if you add a phosphate group, they become on. They get activated. So now let's imagine you discovered a protein that turns on a MAP. Okay? What would you call that protein? It phosphorylates a MAP. Yeah. You might call it a MAP kinase. And in fact, they did call it a MAP kinase. And so these transcription factors are all turned on by a kinase, and the general name for these kinases is MAP kinases or MAPKs. When MAP kinases were discovered, we discovered that they were originally off. They're generally off in the cell, and they can get turned on. And the way that they get turned on is by getting phosphorylated. So now let's imagine you discover a protein that turns on MAP kinase. What might you name the protein that turns on MAP kinase? What do you think, Josh? You're making a face. What do you think? If your answer is silly, that's okay. What would you call it? What would you? It phosphorylates MAP kinase. Yeah, Vicky. Maybe Vicky would call it MAP kinase, and in fact, that's what they called it, because it phosphorylates MAP kinase. MAP kinases, again, are a general class of proteins that work here. When scientists discovered MAP kinases, they found out that MAP kinases are typically inactive in the cell, but they can become activated. And the way you activate a MAP kinase is by phosphorylating it. So if you discovered a protein that activates a MAP kinase, what would you call the protein that activates MAP kinase? Yeah, Josh. You would call it MAP kinase. You can see we're real creative here. And this is, in fact, the name of the general class of proteins that acts here. When scientists discovered MAP kinases, they found that they are traditionally off in cells, and they have to get activated by another protein. The protein that activates them is known as RAS. And the discovery of that was a whole other thing. So, if you remember that the name of this branch is the RAS MAP kinase pathway, hopefully you can remember the details of this pathway, because it's RAS, which turns on MAPKK, which turns on MAPKK, which turns on MAPK, which turns on our transcription factor. And you can see that here. RAS gets activated as a result of LAT and SLP76. Once we have RAS activated, we turn on our MAP kinase cascade, which involves MAPKK, MAPKK, and MAPK. And that turns on our transcription factor and allows our transcription factor to do transcription. So you see, it seems all like hard. It's really not that crazy, not that hard. We can handle this. As I mentioned, there are RAS is just like one protein. But there are multiple examples of MAPKKs. So most commonly, the MAPKK here is called RAF. Wow. Self, you get a lot of demerits for that. It's called RAF. The MAPKK is MEK. The MAPK is ERK. And it turns on a transcription factor named June. Does that say FOS? Man, I'm really off today. It turns on FOS. Bad job, Self. You've only been doing this for like hundreds of years. In other situations, we can get some other MAPKKs, that kind of thing. And sometimes we can also turn on a MAP kinase called JNK, JNK, and it turns on a transcription factor called June. So we often talk about RAS, then RAF, then MEK, then ERK, which turns on FOS. We can also talk about JNK that turns on June. And you can see here's FOS getting turned on this slide. You can also see, oh, June got turned on some other way, right? It was through junk, which stands for actually June kinase. And when Phos and June come together, when Phos and June are both made, when, shall we say, their powers are combined like Captain Planet, they're called AP1. And so you can see Phos plus June together makes this transcription factor called AP1. And so our RAS-MAP kinase pathway will lead to AP1 transcription factor. And that was one of our 3 transcription factors. So if we can kind of remember just RAS-MAP kinase leads to AP1, that's pretty much what you got to remember for that one. So if you remember the name RAS-MAP kinase, that tells you all the people involved. And then that leads to AP1, which is one of our 3 transcription factors. So this branch is relatively straightforward. And then we can go on to our other branch. And so this branch, you can see I have listed as number 2. And it is actually going to kind of branch a little bit and then branch again. Yeah, sorry, Josh.
I'm sorry. So
yep.
from MAP to RAF, is that like?
Oh, it goes from RAS to RAF.
RAS to RAF.
RAS to RAF. RAS, RAF, MECERC. See, I can do it when I do it like that. That is RAS, RAF, MECERC, but yeah. So RAS, then RAF, then MEC, then ERC, then POS. Or RAS, then some stuff that I'm not naming, eventually junk June. Does that answer your question? Okay. And RAS is turned on as a result of our adapter proteins, LAT and SLP76. So our second branch here involves a different protein, though it is also turned on by LAT and SLP76. So now we come back to LAT and SLP76, and instead of going down this RAS branch, we're going to do a branch with a different protein, and that protein is called PLC gamma. One thing that will really help us in thinking about PLC gamma is thinking about what the letters PLC stand for. So PLC, well really PL. PL stands for phospholipase. So you can see phospholipase here, and that's the PL in PLC gamma. It's the phospholipase number C gamma of all the phospholipases that exist. So if we know the name of this protein is phospholipase C gamma, that can tell us something about the protein. So what, if a protein's name is phospholipase, what does that tell us that the protein will do? Yeah. So maybe we think about phosphorylating lipids, but not quite, because then you'd be like, why isn't it kinase? Right? What does ACE sometimes mean? What's a nuclease or what's a protease? Yeah. It cuts things up. So a protease cuts up proteins. A nuclease cuts up nucleic acids. So what do you think a phospholipase might do? Yeah, Vicky. Cuts up phospholipids. Phospholipase C gamma, PLC gamma, doesn't cut every single phospholipid in the whole world, but it cuts one specific phospholipid. So there's one phospholipid that PLC gamma cuts. PLC gamma cuts this phospholipid that's shown here on the left. You don't have to get super stressed about its structure or anything, but I know some of you will, because of your biochemistry background, will be interested in it. This is called PI45 bisphosphate. Most of the time, immunologists refer to it as PIP2. And so this is PIP2. And phospholipase C cleaves PIP2. And when phospholipase C cleaves PIP2, one thing that you should first notice about PIP2 is it has these two long tails, and those are usually close to the cell membrane. And it also has this head group. And when PLC gamma cuts PIP2, it cuts it into two pieces. One of them is mostly the tails. It's called 1,2-diacylglycerol. I usually refer to it as DAG. And the other one is the head group. The head group is inositol 1,4,5-triphosphate, also known as IP3. And so the idea here is that PLC gamma is going to cut PIP2 into DAG and IP3. If you recall, I told you that we're going to have our branch number 2 have like a 2A and a 2B. One of them is going to be where we're going to follow what happens to IP3. The other one is going to be that we follow what happens to DAG. And so we've already got one transcription factor, AP1. We need 2 more. One of them is going to come from IP3. One of them is going to come from DAG. So there is when IP3 is made, because of this cutting, it's going to go bind to another protein in the cell. In some ways, you can think of that as like a receptor, right? That's in a different part of the cell. If you were going to name this protein, what would you name this protein? It's going to bind IP3 kind of like a receptor. Yeah, Vicky. Vicky would name it IP3 receptor, and Vicky is an excellent signal transduction namer. Because in fact, IP3 will then go bind to the IP3 receptor. The IP3 receptor sits on the endoplasmic reticulum, and the end result is that calcium that was in the endoplasmic reticulum gets released into the cytoplasm. You can see this is so here it's just kind of shown like very broadly. This is a figure that shows the same thing in slightly more detail. So this little yellow thing is not labeled, but it's the IP3 receptor. Here's PLC gamma. It's cleaving my phospholipid, PIP2, to make DAG and IP3. IP3 binds to the IP3 receptor. Normally, the cell is super organized. I'm trying to think of the right word. Super strict in terms of what it does with calcium. Normally, the cell has absolutely no calcium in the cytoplasm. If there's any calcium around, the cell puts it in a specific location, generally the ER. So you can see the ER has lots of calcium. See it in blue dots. And the cytoplasm has no calcium, no blue dots. When IP3 binds to the IP3 receptor, suddenly we just open the gate and let the calcium out of the ER. And it just goes from high concentration to low concentration, kind of simple process, when we open that calcium channel. And suddenly we flood the cytoplasm with calcium. And this is a big change in the cytoplasm. You know, you have one molecule of IP3, you get a ton of calcium. And so it like really amplifies the signal. Sometimes we talk about this as a second messenger. If you guys have taken a lot of neuroscience classes, calcium signaling, kind of similar to this, is actually quite important in a lot of neuroscience contexts as well, though we're only talking about it in this T-cell context here. Now, when there's a bunch of calcium in the cell, the calcium there are proteins that are calcium binding proteins. And so you can see that calcium that's now in the cytoplasm can bind to a calcium binding protein like calmodulin, which this textbook has decided looks like a worm. So you can see calcium binds to calmodulin. Calmodulin then undergoes a conformational change and will bind to its target. You can also see the same thing here. Here is my calcium. It's in the ER. The cytoplasm has no calcium. When we open the channel, because of IP3 binding to the IP3 receptor, calcium floods into the cytoplasm and binds to calmodulin. This textbook has decided it's a wrench and not a worm. So you can see now we get binding of the calcium to calmodulin. We get this conformational change. It turns into a wrench. And this allows it to activate later proteins, particularly one called calcineurin. And so you can see calcineurin gets activated. Now calcineurin can do its thing. And calcineurin's thing is that calcineurin removes phosphate groups from its target protein. The target protein is NFAT. NFAT, when it has phosphates on it, is not allowed into the nucleus. So the nucleus is like, no, you're not allowed in here. You can't come in if you have phosphates. When calcineurin is active, it pulls all the phosphates off NFAT, and now NFAT is allowed into the nucleus to do transcription. NFAT was actually named nuclear factor of activated T cells because it's a transcription factor in T cell activation. And it is the second of our transcription factors we had to get to. If you ask many immunologists, in general, this NFAT calcium pathway is the one that they will know the best of all of these T cell activation pathways. And the reason why immunologists will know this pathway the best is because we have figured out drugs that modify this pathway. And so if we have patients who have too much T cells, their T cells are causing them a problem, we can give them a drug that stops this calcium pathway, and then NFAT cannot go to the nucleus and get turned on. And then we can only get 2 of our 3 transcription factors, so the T cells are out of luck. The T cells cannot get all 3 transcription factors they need to get turned on. This drug is called cyclosporin A. And so here you can see cyclosporin A, which is shown in orange. If cyclosporin A, this drug is around, it gets in between calmodulin and calcineurin. So basically all the earlier steps, the LCK, the ZAP-70, the LAT, PLC gamma, all that happens. We release the calcium, and then we can never actually turn on calcineurin. So NFAT never loses its phosphates, and it's never allowed into the nucleus, and the T cell never gets turned on. And this drug is the reason why we do organ transplantation. Organ transplantation did not exist or barely existed before we had this drug to help so many patients' lives. And so because we have this drug, it's sort of the pathway that most immunologists think the most about. At this point, we have turned on 2 of our 3 transcription factors that we needed. And so we need to turn on our 3rd transcription factor. And some people like to talk about just like, and it also happens. I'm going to tell you slightly more than that. Because I always feel bad when I talk about this pathway that I like left out DAG. We cut PIP2 into DAG and IP3, and then we follow IP3's life, and we left poor DAG alone. So what I will just tell you is that DAG turns on its own other signaling pathway that I'm not going to go through in great detail. But DAG eventually turns on our final transcription factor, NF-kappa B. And so once we have DAG being made DAG, while IP3 is off doing the calcium stuff and turning on NFAT, DAG is actually signaling through some other proteins, and turning on our final transcription factor, NF-kappa B. And so in the end, we have all of these things happening at once. So we've got our T-cell receptor binding to MHC plus peptide. We're bringing in our kinase, LCK, on CD4 or CD8. We're phosphorylating our ITAMs, which is giving us new binding sites for ZAP-70. That's allowing more phosphorylation, more binding sites, so LAT and SLP76 can come in. And then we get RAS, turning on the RAS-MAP kinase pathway, and AP1, and PLC gamma, cleaving PIP2 to give us IP3 and NFAT via some calcium, and DAG to give us NF-kappa B. And now, hooray, we have an NFAT, AP1, and NF-kappa B. The cell can be activated. The cell can make IL-2, which was our whole goal in this scenario. So, if you recall, in a past lecture, I told you about the professor I used to TA for, Shiv Pillai, and I showed you a video of his lymphocyte wrap about some details of some immunology. He also wrote a poem about T-cell receptor signaling. Sadly, if you look for this on YouTube, the person's phone ran out of battery halfway through, and so only half of it is on YouTube. But I have the text of the whole thing. And so I am going to read it instead of you getting to watch him read it, which is way less fun than watching him read it, but that's what we got. So that's why I am showing you the T-cell receptor signaling as I read this poem, so you can be following along in the poem. O Hercules and Odysseus, great heroes of the past, we lymphocytes are forsaken. We fear we may not last. Like you, we're xenophobic, and at our very core, like they're against pathogens. That's where against pathogens. We don't like those foreign pathogens. Like you, we're xenophobic, and at our very core, political correctness wholeheartedly deplore. Again, it's about pathogens. You Greeks had mighty heroes, each like a lymphoid clone. They defended the homeland, faced danger all alone. Came a foreign invader, and an army would be raised. 'Twas clonal expansion. To this Zeus be praised. No room for any mercy, that is our battle cry. Self we tolerate gladly, but non-self must surely die. We always had our enemies, but now face gross abuse. Lord, our signals go unanswered. We've got the cyclosporin blues. You see a Phoenician traitor. You know it bodes no harm. For a dreaded Persian worship, you must sound the alarm. Selective xenophobia. No B7 means you're spared. That's the second signal too that we're going to get to later. Second signals spell out danger. For battle, we're prepared. That's also in the homework for Friday. See, when I do it, you get editorial comments. The TCR sees its peptide in an MHC array. Co-receptors bind MHC and bring LCK in play. Those YXXL tyrosines now have phosphates to show. ZAP-70 binds to an ITAM. ZAP-70 hugs an ITAM. The cell is good to go. No room for any mercy. That is our battle cry. Self we tolerate gladly. Non-self must surely die. We always had our enemies, but now face gross abuse. Lord, our signals have gone crazy. Got the cyclosporin blues. ZAP-70 gives PLC a very gentle shove. PLC and LAT are now very much in love. The enzyme releases IP3. It floats into the cell. On its ligand-gated channel, The sugar casts a spell. From the ER and mitochondria, calcium begins to flow. The cell is soon suffused with a calmodulin glow. With calcium calmodulin, calcineurin forms a trio. They samba together like it's carnival in Rio. No room for any mercy, that is our battle cry. Self we tolerate gladly, but non-self must surely die. We've always had our enemies, but now face gross abuse. Oh Lord, we're going crazy, got the cyclosporin blues. That early calcium efflux depletes the ER store. Yeah, we didn't talk about this part, whatever. Calcineurin, like Oliver, asks the cell for more. Stim1 links the ER to the wavy cell membrane, tags ORI the crack channel to join the calcium game. From the surface of NFAT, a phosphate group is lost. No cytosolic retention, the nuclear pore is crossed. The promoters then awaken. The cells begin to churn. From G1S to M phase, the cell cycle will turn. No room for any mercy, that is our battle cry. Self we tolerate gladly, but non-self must surely die. We've always had our enemies, but now face gross abuse. Or Lord, we're going crazy, got the cyclosporin blues. My host has moribund kidneys. An organ was received. Have we not fought with honor? Must we now be deceived? The TCR is activated. A calcium high ensues, but calcineurin lies frozen. We got the cyclosporin blues. NFAT lies frustrated. Promoters fail to churn. The engine keeps on cranking, but the wheels fail to turn. Once we fought with honor, but now in hell we burn. Consecrate our ashes in a drug-drenched hero's urn. No room for any mercy, that is our battle cry. Pathogens we dealt with gladly, but it is now our turn to die. We've vanquished all our enemies, but now we must learn to lose. Open those gates to Hades, we've lived the cyclosporin blues. So, what you will notice there is he got the whole thing. And also it talked about what cyclosporin will do in terms of the upstream parts of the pathway happen, but the cell is unable to move forward and actually complete this process to turn on that final transcription factor. And thus we do not get AP1, NFAT, and NF-κB together. NFAT is missing, and we cannot transcribe IL-2 and make the IL-2 protein. So now that we've made IL-2, we can think about exactly what this protein does that we've made. IL-2 is actually one of the cytokines that can be important for that signal 3. And so IL-2 is a really critical cytokine here. When IL-2 was first discovered, it was named TCGF, or T-cell growth factor. So that should tell you a lot about what IL-2 does. IL-2 basically allows for T-cells to undergo the cell cycle. It's a mitogen. It activates mitosis. And it also really allows the cell to be able to differentiate and survive. So IL-2 is sort of this key growth factor that the T-cell needs in order to become really activated. IL-2 is part of a family of cytokines. And this family of cytokines is known as the common gamma chain family. The reason why it is known as the common gamma chain family is that all of the receptors for cytokines in this family share a chain. They have that chain in common. And that chain is known as the gamma chain. So it's the common gamma chain family. So here you're looking at the receptors for some members of this family. And what you'll notice is they all have this same gamma chain that they pair with other proteins. And so the IL-7 receptor, for example, really involves the gamma chain plus an IL-7 specific chain, an IL-7 receptor. IL-4 you can see similarly, IL-9 you can see similarly. Both IL-15 and IL-2 actually have 3 chains, but they all share this gamma chain. There's also another protein called IL-21 that's not shown here that's in this family. So you've heard of one of these cytokines earlier this semester. So which of these cytokines have you heard of this earlier this semester, and where have you heard of it? Yeah, Kyra. Maybe not. I mean, maybe it shows up in the textbook, but I usually haven't talked about IL-9. Yeah, Emma. IL-7. So we talked a little bit about IL-7 previously. Where did IL-7 show up? Yeah. So we saw it with developing B cells. I didn't really highlight it, but it actually is also in the thymus for developing T cells a lot too. It's really important for sort of early developing lymphocytes. And now you see IL-2, which is like super important for turning on T cells. And in fact, if I were to go through all the members of this family, they're really important for some key aspect of some lymphocyte working. The reason why I mention this is that one particularly common immunodeficiency disease is called severe combined immunodeficiency, or SCID. And the most common form of SCID is when patients are missing the common gamma chain. So when they're missing the common gamma chain, that means they have no IL-2 receptor and can't do IL-2 things. So no turning on T cells. It also means they're missing IL-15 receptor because it's a shared protein, and can't do IL-15 things, which we haven't really talked about yet. Can't do any developing B cells or T cells. That kind of gets rid of all your lymphocytes right there because no IL-7. Also no IL-9 and IL-4, who again we can talk about at a later time. And so this is a very severe deficiency. These kids basically do not have an adaptive immune system. I showed you the boy in the bubble earlier in the semester. He had a case of common gamma chain SCID. The common gamma chain is encoded on the X chromosome, so this is an X-linked disease. Most of the patients with this are boys. IL-2 actually can bind a little bit to just its own receptor, the IL-2 receptor. And you can see we can get kind of low affinity, medium affinity, or high affinity IL-2 receptors. IL-2 binds best when all 3 chains are present, alpha, beta, and gamma. But it binds a little if we haven't quite made alpha yet. And so what will happen is our cell will, at the beginning, have just beta and gamma, and then it will make some IL-2. And guess who it will feed that IL-2 to? It's like if I made some cookies. Who am I going to give my cookies to? Me! I'm going to give them to me because I like cookies. So the cell actually starts making this growth factor IL-2 and feeds it to itself. And so it starts making IL-2, starts feeding it to itself, and the cell's like, hey, this is a good idea. Let me make this really good receptor and even get more cookies. And get even more high affinity of this process. And so here you can see this. Originally, our naive cell doesn't really make any IL-2. It also isn't really making that IL-2 receptor, the high affinity IL-2 receptor. But if we turn on that cell through signal 1, we get our 3 transcription factors, and our cell makes IL-2. And then the cell just feeds itself the IL-2, which it's now really ready to receive. That was also a signal that came through the T cell receptor. And now the cell basically made itself divide because it fed itself this IL-2 and this growth factor. So it makes a growth factor and then it feeds itself growth factor. Usually what really does happen is you might have a bunch of T cells in a small area. And so they're feeding themselves and also their neighbors, but it's also kind of going around in just that small area. It's not like all the T cells in the whole body get IL-2. It's just this sort of autocrine and paracrine process. And so that's kind of the point of IL-2. I need to tell you a little bit more about signals that happen downstream of cytokine receptors. So we're going to have another signaling pathway. It's kind of like RAS-MAP kinase, that if you know the name, you know the pathway. So we'll talk about that on Friday, and then we'll kind of move into the next piece of T-cell activation. And remember that PRR assignment is due on Friday. It will tie very tightly into the lecture material that's coming up on Friday.